Hi, I'm Jiale. In this video, I'll be talking through data from our recent paper published in the Journal of Immunology titled B cells from aged mice do not have intrinsic defects in affinity maturation in response to immunization. The full paper can be accessed via the QR code shown on the slide as well. Affinity maturation refers to the phenomenon that results in the progressive increase in affinity of serum antibodies over time. It is therefore an important process that underlies an effective vaccine response. Germinal centers, or GCs, are the cellular engines of affinity maturation. Within GCs, B cells undergo somatic hypermutation of their antibody genes in the dark zone. Due to the random nature of the mutation process, B cells will then undergo selection in the light zone, where B cells that can uptake and present antigen to TF8 cells will receive positive selection signals. These B cells upregulate the proto-oncogene c-MYC, which promotes re-entry into the dark zone for further somatic mutations and clonal expansion. Alternatively, B cells that fail to receive positive selection signals will undergo apoptosis. Eventually, these B cells exit the GC as memory B cells or long-lived plasma cells, which are key in conferring protection against future infections. Previous studies from our lab and other labs have shown that affinity maturation is impaired during aging following immunization. For example, GC B cells in aged mice have a near twofold reduction in the frequency of the affinity-enhancing W33L mutation compared to those in young mice following immunization. In addition, a reduction in the ratio of high-affinity MP2 binding to MP23 binding plasma cells is also observed in aged mice following immunization compared with young adult mice. Since B cells are the key cell type that mediates humoral response, we aim to investigate in this paper if B cells from aged mice have intrinsic defects in undergoing affinity maturation following immunization. In particular, we looked at their ability to accumulate mutations, receive positive selection signals, and develop into high-affinity antigen-specific clones. To interrogate this question, we performed adoptive transfers with B cells from B1AI donor mice. B1AI mice are transgenic mice with B cells that contain the knock-in heavy chain gene that confers an intermediate affinity for the MP hapten. For our study, we transferred MP-specific B cells from 6 to 12 weeks old young adult mice, or aged mice that are more than 90 weeks old, into young congenically distinct wild-type mice. We then tracked the B cells' response at different time points post-immunization with MPKLH in alum in draining inguinal lymph nodes of recipient mice. Prior to the transfer, we stained the donor B cells from young adults or aged mice with a comprehensive flow cytometry panel to check for phenotypic differences that might affect B cell responses to immunization. Figure 1a shows the gating strategy for the different subsets of B cells analyzed among MP-binding B cells, namely follicular B cells that are CD23 high, CD21 intermediate, marginal zone B cells that are CD23 negative, CD21 positive, and CD23, CD21 double negative B cells. We then enumerated the percentages of these subsets in Figure 1b. Aged B18 mice had on average 10% fewer IgG-positive naive MP-binding B cells and follicular B cells compared to young adult mice. Correspondingly, there was a tendency for aged mice to have higher proportions of marginal zone B cells, double negative B cells, and CD11c positive atypical B cells. These age-related effects on B cell subsets were also observed in the total B cell population, which we show in supplementary Figure 1. We also assessed the expression levels of various proteins on the MP-binding B cells from young adults and aged mice, and generally found no age-related differences. These proteins include chemokine receptors and trafficking molecules CXCR4, CD62L, and CXCR5, B cell activation markers CD8, GL7, and IR4, atypical or age-associated B cell markers CXCR3, CD11b, and CD11c, and co-stimulatory molecules CD86, CD40, and MHC class II. Furthermore, when we gated the total MP-binding B cells into individual B cell subsets and analyzed expression levels of these markers, no significant age-dependent differences were observed, as shown in supplementary figure 2. These are the MFI of the markers for MP-binding follicular B cells, marginal zone B cells, and double-negative B cells. Together, this shows that B cells from aged B18 mice are generally phenotypically similar to those from young adult mice. Next, to determine if there are any cell intrinsic defects by B cells from aged mice in responding to immunization, equal numbers of MP-specific B cells from either a young adult or aged B18 mouse were adoptively transferred into young congenic wild-type recipient mice, as previously described. Their responses were assessed in draining inguinal lymph nodes of recipient mice at days 6, 10, 14, 21, and 28 post-immunization with MPKIH in alum. As shown in figure 2b, across all time points, there was no significant difference in the percentage and number of MP-specific B cells from the young adult or aged donor recovered in the recipient lymph nodes, suggesting no age-related defects in the ability of B cells to survive, migrate, and/or proliferate in the recipient mice. Across all time points, we also observed no age-related cell intrinsic defects in the ability of MP-specific B cells from aged mice in class switching to IgG1, or in becoming GC B cells identified as CD38 negative GL7 positive cells. There was also no significant difference in the percentage and number of short-lived extrafollicular plasma cells derived from donor cells from aged mice compared to those from the young adult mice. Together, this shows that there are no cell intrinsic defects in the ability of MP-specific B cells from aged B18 mice in responding to stimulation and differentiating into plasma cells or GCB cells. Next, we compared the ability of B cells from young adults or aged mice in undergoing positive selection, which primarily occurs in the light zone of the GC. Using the aforementioned adoptive transfer system, we compared the ability of B18 B cells to upregulate c-MYC at the peak of the GC response, which is day 6 post-immunization. Shown in figure 3a is the gating strategy we used to assess c-MYC expression in the donor B cells, which carry a c-MYC GFP reporter gene. The host B cells serve as a negative control since they do not carry the c-MYC GFP reporter gene. The c-MYC positive GCB cells were predominantly of the light zone phenotype as shown in figure 3b. MP-specific GCB cells and light zone GCB cells derived from aged mice also showed no intrinsic defects in c-MYC upregulation, as shown in figure 3c and d. This suggests that aging does not diminish the ability of B cells to receive positive selection signals in a young microenvironment. We then compared the ability of B cells from aged mice to undergo somatic hypermutation and accumulate affinity-enhancing mutations. To do so, we sequenced the VH166.2 region of sorted donor-derived MP-specific GCB cells at day 14 post-immunization. The W33L mutation, which is a substitution mutation at position 33 of the CDR region of VH16.2, results in a tenfold increase in antibody affinity for MP. We observed in figure 4a that there was no significant difference in the frequency of W33L mutation among sorted MP-specific GCB cells from young adult and aged donor mice. And this is enumerated per mouse in the graph shown in figure 4b. In addition, the number of mutations at the VH186.2 site and the ratio of replacement to silent mutations were similar among GCB cells from young adult and aged mice. Together, this suggests that B cells from aged mice are equally functional in undergoing somatic hypermutation and receiving positive selection signals, and ultimately producing GCB cells with affinity-enhancing mutation in a young microenvironment. Since we have observed that B cells from aged mice were capable of undergoing affinity maturation, we hypothesized that B cell extrinsic factors contribute to the age-related impairments observed after immunization. To test this, MP-specific B cells from young adult B18 mice were adoptively transferred into young or aged congenic wild-type recipient mice, as shown in Figure 5a, and the B-cell response was analysed at day 14 post-immunisation. As shown in Figure 5b, MP-binding B cells were able to participate in the GC response in both adult and aged recipient mice, although the number of GC B cells was reduced in the aged mice. To assess somatic hypermutation and acquisition of high-affinity variants, we similarly performed sequencing of the VH186.2 region of sorted donor-derived GC B cells in young or aged hosts. Sorted MP-specific GC B cells from young adult donor mice showed significant defects in their ability to acquire high-affinity mutations when transferred into aged recipient mice, compared to those transferred into young mice, as shown in Figures 5c and d. Furthermore, donor-derived GC B cells in aged recipient mice had significantly fewer mutation in the VH186.2 region and lower ratio of replacement to silent mutations than those in young recipients. Together, this shows that an aged microenvironment is a key contributor to defects in affinity maturation upon immunization. To summarize the findings from this paper, we first observed that B cells from aged B18 mice are less frequently of a follicular phenotype. Despite that, they have similar expression levels of various markers. Through our kinetic studies, we show that B cells from aged B18 mice do not have intrinsic defects in class switch recombination, entering the GC response and plasmablast differentiation upon transfer into young recipient mice. In addition, GC B cells from aged mice are able to receive positive selection signals and upregulate c-Myc, and accumulate high-affinity mutations post-immunization in young mice. Conversely, B cells from young mice have fewer high-affinity mutations when transferred into aged hosts, implicating B-cell extrinsic factors as key contributors to defects in affinity maturation upon immunization. I would like to thank all the co-authors who contributed to this study, as well as support from the Babraham Institute Flow Cytometry Facility and Biological Support Unit. We would also like to thank our funding bodies for their support, and thank you for your attention.